Welcome friends, welcome to the Knowledge Club Online. Now I am going to discuss about the tertiary structure of DNA. DNA helix can be deformed from their linear shape into curves and bends. Real DNA does this as a result of normal thermal motion and under the influence of certain proteins which bind to DNA. The nucleosome, which is a DNA packing structure, is one example where DNA is held in a non-linear form. Each nucleosome consists of a core of histone proteins around which the DNA double helix is wrapped, some 1.6 turns. Other proteins, such as DNA transcription factors, also alter the geometry of DNA and thereby modify its ability to be transcribed into RNA. This is a model, the supercoiling. Now, when the DNA helix has the normal number of base pairs per helical turn, it is in the relaxed state. Changing this normal amount of twist can be demonstrated by grasping both ends of the short linear model, 1 to 2 complete turns, and twisting the ends in opposite directions. If the helix is over twisted so that it becomes tighter, the edges of the narrow groove move closer together. If the helix is under twist, the edges of the narrow groove move further apart. Notice that changing the twist from the relaxed state requires adding energy and increases the stress along the molecule. DNA double helices can fold up on themselves to form tertiary structures created by supercoiling. This is the picture of supercoiling. If DNA is in the form of a circular molecule, or if the ends are rigidly held so that it forms a loop, when over twisting or under twisting leads to the supercoiled state. Supercoiling occurs when the molecule relieves the helical stress by twisting around itself. Over twisting leads to positive supercoiling, while under twisting leads to negative supercoiling. Most DNA molecules inside cells are subject to supercoiling. Supercoiling markedly alters the overall form of DNA. A supercoiled DNA molecule is more compact than a relaxed DNA molecule of the same length. Supercoiled DNA moves faster than relaxed DNA when analyzed by centrifugation or electrophoresis. Although supercoiling relieves some stress in the molecule when there is negative supercoiling, further stress can be relieved by partial strand separation. The hydrogen bonds holding together complementary bases break and part of the double helix separates. Strand separation is required for transcription, copying DNA to RNA, and replication, copying DNA to DNA. In the figure below, the yellow strands have separated, allowing the molecule to relax and assume a structural configuration. The models above are built with white rods for the base pairs and small green rods for the phosphodiesterase bonds. Yellow 180-degree connectors were used for the separating strands. The models contains approximately 26 helical turns or 160 base pairs. Now, the linking number of DNA, a topological property, determines the degree of supercoiling. In example, a linear 260 BP DNA duplex in the bDNA form. Because the number of base pairs per turn in an unstressed DNA molecules averaged 10.4, This linear DNA molecules has 25 to 60 per 10.4 turns. The ends of this helix can be joined to produce a relaxed circular DNA. A deformed circular DNA can be formed by unwinding the linear duplex by 2 turns before joining its end. 2 limiting conformations are possible. The DNA can fold into a structure containing 23 turns of B helix and an unwound loop. Alternatively, the double helix can fold up to cross itself. Such crossing are called supercoils. In particular, a supercoiled structure with 25 turns of B helix and 2 turns of right-handed turn, where a key topological property of a circular DNA molecule is its linking number Ln, which is equal to the number of times that a strand of DNA winds in the right-handed direction around the helix axis when the axis is constrained to lie in a plane. For the relaxed DNA shown in figure, Ln is equal to 25. For the partly unwound molecule shown in part D and the supercoiled one shown in part E, Ln is equal to 23 because the linear duplex was unwound to complete turns before closure. Molecules differing only in linking number are topological isomers, topoisomers of one another. Topoisomers of DNA can be interconnected only by cutting one or both DNA strands and then rejoining them. The unwound DNA and supercoiled DNA shown in figure D and E are topologically identical but geometrically different. They have the same value of Ln but differ in twist Tw and width Wr. Twist is a measure of the helical unwinding of the DNA strand around each other. Width is a measure of the coiling of the axis of the double helix, that is supercoiling. A right-handed coiler is assigned a negative number, negative supercoiling, and a left-handed coil is assigned a positive number, positive supercoiling. Topology tells us that the sum of Tw and Wr is equal to Ln. Ln is equal to Tw plus Wr. In figure, the partly unwound circular DNA has Tw 23 and Wr 0, whereas the supercoiled DNA has Tw 25 and Wr 2. The free energy is minimized when about 70% of the change in Ln is expressed in Wr and 30% is expressed in Tw. The most stable form would be 1 with Tw is equal to 24.4 and Wr is equal to minus 1.4. Thus, the lowering of Ln causes both right-handed negative supercoiling of the DNA axis and unwinding of the duplex. Topoisomers differing by just 1 in Ln and consequently by 0.7 in Wr can be readily separated by agarose gel electrophoresis because their hydrodynamic volumes are quite different. Supercoiling condenses DNA. Now I come to the topic of the topoisomers. Prepare the double helix for unwinding. Most naturally occurring DNA molecules are negatively supercoiled. Negative supercoiling arises from the unwind or underwinding of the DNA. In essence, negative supercoiling prepares DNA for processes requiring separation of the DNA strands, such as replication. Positive supercoiling condenses DNA as effectively, but it makes strand separation more difficult. Topoisomers are enzymes that regulate the overwinding or underwinding of DNA. The winding problem of DNA arises due to the intertwined nature of its double helical structure. During DNA replication and transcription, DNA becomes overwound ahead of a replication fork. It left unabated. This tension would eventually stop the ability of the enzymes involved in this process to continue down the DNA strand. In order to prevent and correct these types of topological problems caused by the double helix, topoisomers bind to either single-stranded or double-stranded DNA and cut the phosphate backbone of the DNA. This intermediate break allows the DNA to be untangled or unwound, and at the end of this process, the DNA backbone is resealed again. Since the overall chemical composition and connectivity of the DNA do not change, the tangled and untangled DNAs are chemical isomers, differing only in their global topology, thus their name. Topoisomers are isomers enzymes that act on the topology of DNA. Bacterial topoisomers and human topoisomers proceed via the same mechanism for replication and transcription. The double helical configuration that DNA strands naturally reside makes them difficult to separate, and yet they must be separated by helicase enzymes. If other enzymes are to transcribe the sequences that encode proteins, or if chromosomes are to be replicated. In so-called circular DNA, in which double helical DNA is bent around and joined in a circle, the two strands are topologically linked or knotted. Otherwise, identical loops of DNA having different numbers of twists are topoisomers and cannot be interconverted by any process that does not involve the breaking of DNA strands. Topoisomerase catalyze and guide the unknotting or unkinning of DNA by creating transient breaks in the DNA using a conserved tyrosine as the catalytic residue. In this section describes the two different classes of topoisomers and their role in DNA replication. Type 1 topoisomers catalyze the relaxation of supercoiled DNA, a thermodynamically favorable process. Type 2 topoisomerase utilizes the free energy from ATP hydrolysis to add negative supercoils to DNA. These enzymes alter the linking number of DNA by catalyzing a three-step process. First, the cleavage of one or both strands of DNA, the passage of a segment of DNA through this break, and the releasing of the DNA break. There are three main types of topology: supercoiling, knotting, and catenation. Outside of the essential process of replication or transcription, DNA must be kept as compact as possible, and these three states help this cause. However, when transcription or replication occurs, DNA must be free, and these states seriously hinder the processes. In addition, during replication, the newly replicated duplex of DNA and the original duplex of DNA become intertwined and must be completely separated in order to ensure genomic integrity as a cell divides. As the transcription bubble proceeds, DNA ahead of the transcription fork becomes overwound or positively supercoiled, while DNA behind the transcription bubble becomes underwound or negatively supercoiled. As replication occurs, DNA ahead of the replication bubble becomes positively supercoiled, while DNA behind the replication fork becomes entangled, forming precatenance. One of the most essential topological problems occurs at the very end of replication, when the some chromosomes must be fully disentangled before mitosis occurs. Topoisomerase 2A plays an essential role in resolving these topological problems. Type 1 topoisomerase cleave just one strand of DNA, whereas type 2 enzymes cleave both strands. First, describing the type 1 topoisomerase relax DNA by nicking and then closing one strand of duplex DNA. And topoisomerase 1 comprises 4 domains: which are arranged around a central cavity having a diameter of 20 Armstrong, just the correct size to accommodate a double-stranded DNA molecule. This cavity also introduces tyrosine residue, which acts as a nucleophile to cleave the DNA backbone in the course of analysis. The first topoisomerase to be discovered, E. coli topoisomerase 1, can remove negative supercoils without leaving nicks in the DNA molecule. After the enzyme binds to a DNA molecule inside the cavity of topoisomerase, the hydroxyl group of tyrosine 723 attacks a phosphate group on one strand of the DNA backbone to form a phosphodiester linkage between the enzyme and the DNA. It cuts one strand and released 5' hydroxyl group on the DNA. With the backbone of one strand cleaved, the DNA can now rotate around the remaining strand, it a movement driven by the release of energy stored because of the supercoiling. The rotation of the DNA unwinds the supercoils. The enzyme controls the rotation so that the unwinding is not rapid. Formation of this phosphotyrosine bond does not require ATP or another source of energy. The free 3' hydroxyl end of the DNA is held non-covalently by the enzyme. The DNA strand that has not been cleaved is then passed through the single-stranded break. The cleaved strand is then released, forming a structure with the same chemical bonds as the starting DNA, but with one less negative supercoil. By this mechanism, the enzyme removes one negative supercoil at a time. Reversible cleavage of one strand of supercoiled DNA allows controlled rotation to partly relax the supercoils. Any enzyme that cleaves only one strand of a DNA duplex and then recedes it is classified as a type 1 topoisomerase, topo 1. The topo 1 from E. coli acts on negative but not positive supercoils. In contrast, topo 1 enzymes from eukaryotic cells can remove both positive and negative supercoils because the relaxation removal of DNA supercoils by topo 1 is energetically favorable. The reaction proceeds without an energy requirement. The sequential action of E. coli topo 1 can remove essentially all the supercoils in a DNA molecule. Gene targeting knockout strains of E. coli have established that topo 1 is essential for viability. The enzyme is thought to help maintain the proper superhelical density of the E. coli chromosomes by removing negative supercoils formed by action of type 2 topoisomerase. DNA guidance, which is discussed later. Since E. coli topo 1 cannot remove positive supercoils, it is unlikely to play a role in a growing fork progression, which generates positive supercoils in yeast, either topo 1 or topo 2, both of which can relax positive and negative supercoils functions in the movement of growing forks. Mutations in the gene encoding topo 1 affect the growth rate of yeast cells but are not lethal. Thus, this enzyme is not essential for viability. However, is topo 2, which separates the two DNA duplexes following replication, is essential for viability. In both yeast and E. coli, topo 1 enzymes may be important in regulating aspects of transcription as well as in repairing damaged DNA. Now, I come to the type 2 topoisomerase changes DNA topology by breaking and rejoining double-stranded DNA. The first type 2 topoisomerase, topo 2, to be described was isolated from E. coli and named DNA guidase. Topo 2 enzymes have the ability to cut both strands of a double-stranded DNA molecule, pass another portion of the duplex through the cut and reseal the cut in a process that utilizes ATP. Depending on the DNA substrate, the markers will have the effect of changing a positive supercoil into a negative supercoil or of increasing the number of negative supercoils by 2. The topo 2 enzymes from mammalian cells cannot, like E. coli DNA gyrase, increase the superhelical density at the dense of ATP. Presumably, no such activity is required in eukaryotes since binding of histones increases the potential superhelicity. All type 2 topoisomerase catalyze catenation and decatenation, that is, the linking and unlinking of two different DNA duplexes. DNA gyrase is composed of two identical subunits. Hydrolysis of ATP by gyrases and ATP's activity powers the conformational changes that are critical to the enzyme's operation. The enzyme functions to introduce negative supercoils at or near the ORC site in the DNA template, as noted earlier. DNA A can initiate replication only on a negatively supercoiled template. Measurements of the degree of DNA supercoiling in E. coli suggest that there is one negative supercoil for each 15 to 20 turns of the DNA helix. A second crucial function of gyrase is to remove the positive supercoils that form ahead of the growing fork during elongation of the growing strands. The topoisomerase 2 from yeast is a dimer with two internal cavities. The larger cavity has gates at both the top and the bottom that are crucial to topoisomerase action. The reaction begins with the binding of one double helix, here referred G, to the enzyme. Each strand is positioned next to a tyrosine residue, one from each monomer capable of forming a covalent linkage with the DNA backbone. This complex then loosely binds a second DNA double helix, here referred T. Each monomer of the enzyme has a domain that binds ATP. This ATP binding leads to a conformational change that strongly favors the coming together of the two domains. As these domains come closer together, they trap the bound T segment. The conformational change also forced the separation and cleavage of the two strands of the G segment. Each strand is joined to the enzyme by a tyrosine phosphodiester linkage. Unlike the type 1 enzyme, the type 2 topoisomerase hold the DNA tightly so that it cannot rotate. The T segment then passes through the cleavaged G segment into the large central cavity. The ligation of the G segment leads to the release of the T segment through the gate at the bottom of the enzyme. The hydrolysis of ATP and the release of ADP and orthophosphate allow the ATP binding domains to separate, preparing the enzyme to bind another T segment. The overall process leads to a decrease in the linking number by 2. Now, the replicated circular DNA molecules are separated by type 2 topoisomerase. During DNA replication, the parental strands remain intact and retain their superhelicity. This poses static and topological constraints to the completion of replication by a circular molecule as the two growing forks approach each other. A similar situation arises, however, two growing forks in a linear eukaryotic chromosome approach each other. Figure illustrates how the first few helical turns in the parental DNA could be removed by changing the topology of the already replicated regions, leaving the two nearly complete daughter helix linked together as catenons, covalently linked but not yet completely finished circles. Replication then could be completed before or after decatenation to yield to separate from daughter helices. In E. coli, decatenation is catalyzed by DNA gyrase and a second type 2 enzyme called topoisomerase 4. which genetic studies suggest is responsible for separating newly replicated molecules in vivo. Furthermore, temperature-sensitive topo4 mutants carrying plasmid accumulate interlocked plasmid, that is, catenons of plasmid DNA, which appear similar to the SV40 catenon shown in figure. To separate, DNA catenons topo4 presumably binds to the interlocked duplexes and makes a double-stranded break in one molecule while remaining attached to the substrate. It then passes the other molecule through the break and finally receives the break in the cut molecule. Interestingly, although DNA guides can carry out decatenation in vitro, it cannot fully substitute for topo4 in vivo, as demonstrated by the lethal effects of topo4 mutations. Now, the linear daughter chromatids also are separated by type 2 topoisomerase. The use of temperature-sensitive mutations in topo2 has shown the importance of this protein in yeast cell viability. Furthermore, light microscopy studies have revealed the participation of topo2 in separating the linear chromosomes of yeast cells. Although individual yeast chromosomes are too small to be visualized by light microscopy, a structure called the nuclear body, which contains all the chromosomes clumped together, can be seen. When temperature-sensitive topo2 mutants are shifted to a non-permissive temperature, the nuclear body, which usually divides at the junction of the mother and daughter cell, appears to get stuck in the passageway between the two cells. Fluorescent body staining of metaphase chromosomes reveals that topoisomerase 2 is the principal component of the non-histone protein scaffolding to which long DNA loops are attached. This finding and the genetic studies in yeast strongly suggest that eukaryotic topo2 resolves tangles that exist in newly replicated linear chromosomes. And finally, the similarity between the phenotypes of topo2 mutants in yeast and topo4 mutants in bacteria argue that control of topological domains may be analog in eukaryotic chromosomes and in small circular DNA molecules. The insertion of viral DNA into chromosomes and other forms of recombination can also require the action of topoisomerases. Many drugs operate through interference with the topoisomerases. The broad-spectrum fluoroquinolone antibiotics act by disrupting the function of bacterial type 2 topoisomerases. These small molecule inhibitors act as efficient antibacterial agents by hijacking the natural ability of topoisomerase to create breaks in chromosomal DNA. Novoviracin blocks the binding of ATP to guidase. Nalidixic acid and ciprofloxacin, in contrast, interfere with the breakage and rejoining of DNA chains. These two guidase inhibitors are widely used to treat urinary tract and other infections, including those due to Bacillus anthracis. Some chromatographic drugs called topoisomerase inhibitors work by interfering with a mammalian type eukaryotic topoisomerase in cancer cells. This induces breaks in the DNA that ultimately lead to programmed cell death, apoptosis. This DNA damaging effect outside of its potential curative properties may lead to secondary neoplasm in the patient, camptothecin example. This lecture may be helpful to you. Thank you.